and welcome everybody to my channel Omni Bioentrance, and today we will be discussing CSIR Net JRF Life Science question paper part C questions of shift 1 from July, conducted in the year July 2024. If you are new to my channel, please do like, share and subscribe. If you want still previous year question papers of CSIR Net JRF, then you can go to my channel Omni Bioentrance, select the playlist. In the playlist, C for CSIR Net JRF, there you will find. Now first question. The CDS of that is the coding sequence. Okay, CDS is coding sequence of the shortest isoform of human gene A is cloned into a 3.3 kb vector under the CMV promoter at the BAM-H1 and XHO sites. Okay. So these are the bands that you obtained. This is the PCMB digested with XHO and BAM-H1. Now Western blot against the A null HeLa cell lysate. Now this is the Western blot that you obtained after you transfect the vector, that is PCMBA, with HeLa cells. Sorry, after you transfer HeLa cells with the vector PCMVA. Now Western blot analysis, you can see a range of multiple bands of higher molecular weight, whereas in the untransfected cell you don't see them, because untransfected cell doesn't have this gene which is coding for protein A. Then, from the agarose gel and SDS page images shown above, which one of the following is most likely a protein for A in HeLa cells? So, protein A form homomultimers. If that was the case, what is a homomultimer? Homomultimer, it is a protein complex where subunits are identical. That is 100% sequence identity. They may be trimers, tetramers, pentamers, hexamers, etc. And they will give only one single band. Okay. So this is not a homomultimer. But because you can see a broad range of high molecular weight markers. So A is not correct. Protein A is degraded by the lysosome. If it had undergone lysosomal degradation, then you would have got smaller bands, not high molecular weight bands. So 2 is also not correct. Protein A is polyubiquitinated. That would be the answer, because polyubiquitination, it causes the protein to have a range of molecular weights because of the addition of ubiquitin chains. So that could be the answer. Protein A delocalizes to autophagosomes. Again, lysosomal degradation where you will get smaller bands. So that is also not correct. So answer here is option 3. Then next one. A plant breeder introduces a gene for pathogen resistance from wild species B. So B is the donor species, into a cultivated variety A. A is the recipient or the recurrent species. In panel 1, in the figure shows a profile of DNA markers for A and B. Panel 2 shows a genetic map for the linkage group which has the gene for pathogen resistance. This R is the pathogen resistance. Which of the following options has the correct choice of markers for foreground and background selection respectively? Now what is this foreground selection? That is when a breeder selects plants that have marker alleles of the donor parent at the target locus. Okay, of the donor parent at the target locus. So of the donor means of wild species B, I told you it is the donor parent. So it will have, this is the resistance gene. So it will have B3 as well as B2. That is foreground selection. Now what is background selection? Background selection is just somewhat opposite, I can say, of that, where the breeder select plants that have marker alleles of the recurrent parent. Here the recurrent parent is cultivar variety A in all the genomic region except the target locus. So it can be A2, it can be A2, it can be A4, it can be A7. So answer here is option 3. Okay, A2, A3, okay, A3, A4 and A7, B3 and B2. Now next one. Cystic fibrosis is caused by a recessive allele. Roughly 1 out of every 500 individuals have this disease. Using Hardy-Weinberg equation, the percentage of individuals who are carriers of the recessive allele for the disease. Now carriers means heterozygous individual, that is 2pq. So q square they have given 500. So 1 by 500, q is equal to 0.04. P is 1 minus q, that is 0.96. 2pq you have to find. 2 into 0.96 into 0.04 will give you percentage of carriers as 7.6. Answer is option D. Now in a frog species, foot webbing is controlled by a single gene where the allele for non-webbed foot W is dominant and webbed feet small w is recessive. Assume there is a population of 500 individuals where 320 have genotype capital W, 160 have the heterozygous genotype capital W and 20 have genotype small w. What are the frequencies of the 3 genotypes and alleles in the population? Answer is option 3. I have solved. So total number of individual is 500. Capital W is 320. Capital W is 160. And small w is 20. All this is given in your question. Then frequency of WW, that is P square, that is number of individuals who are WW by total number of individuals, that is 320 divided by 500, that is 0.64. Then frequency of small w capital W, that is 2pq is 160 divided by 500, that is 0.32, and frequency of q square, that is small w is 20 by 500, that is 0.04. Now allele frequencies. For P it is 2 into 320 plus 160, that is of this is of capital W plus 160 is of capital W small w divided by 2 into 500, you will get it as 0.8. Q is 1 minus P, that is 0.2. So that's it. Thank you for watching my channel. Please do like, share and subscribe if you are new to my channel. If you want still previous year question papers of CSIR JRF been answered, you can go to my channel Omni Bio Entrance. Select in CSIR JRF in the playlist. You will find it there. Thank you. Recessive mutation in the human dysferlin gene lead to limb girdle type 2 muscular dystrophy. The gene is located on the second chromosome. The patient's parents do not have limb girdle type 2 dystrophy. What is the probability that at least one of the 4 grandparents of the patient suffered from this disease? So this is the patient, he or she is suffering from the limb girdle type 2 dystrophy. So he is A. Now his parents, they are not having the disease, so they are carriers. That is why they contributed small a each to the patient and he became deceased. So these are the parents. Now what about the grandparents? At least one of the 4 grandparents suffered from the disease. Now if they are capital A, small a, capital A, small a, then the probability of the grandparent being affected is 1 by 4. Here 1 by 4. Okay. And probability of them being a carrier is half. And probability of not being affected or a carrier is 1 by 4. Probability that neither grandparent is affected is 1 by 4 plus half. 1 by 4 plus half, that is 3 by 4, that is 81 by 256. Probability that at least one grandparent is affected is 1 minus 81 by 256, that is 175 by 256, that is equal to 7 by 10. So the answer here is option 4, 7 by 10.